So good morning, everyone. I'm Uyen Mai, a postdoc at Princeton University. Today, I present my work as a work of my PhD work at UCSD. The title is MDCAT, Phylogenetic Dating under a Flexible Categorical Model using Expectation-Maximization. So we have listened to many fantastic talks yesterday and today about inferring the phylogenetic trees. My talk will be going one step beyond that, where we add the temporal axis into the picture. So we look at the time tree, or also called the chronogram, which is a tree structure that show both how and when the species evolve through time. So from this, we not only can answer how the species or the biological entities are related evolutionary, but also we can know when the speciation events or some important evolutionary event happened in the past. Time trees are needed in many analyses, just as biogeography, ancestral state reconstruction. We can study, use it to study epidemics, the spread of some viruses, or phylodynamics. So in modern biology, we want to infer everything from molecular sequences. And we would like to do that for time tree as well. So molecular dating is the problem of inferring time tree from a set of molecular sequences together with a small set of calibration points from fossil records and infer the time tree out of it. Molecular dating, however, is challenging because when we look at the molecular sequences, we don't have any timing information in it. All we can do at the best is to infer how many mutations or substitution events has happened between the two molecular sequences. But that distance is a product of two unknowns, the time and the mutation rate. So in other words, rate and time are inseparable. And in order to infer the time tree, we need both of them. That rate, actually, if we assume that the rate is constant through time, basically, it's just a proportional between the two metrics. But in reality, the rates actually can be different among the lineages. So the generative model, the probabilistic model here, is comprised of the two stochastic processes. The first one, molecular clock model, it describes the stochastic change of the substitution rate. And the second model is sequence evolutionary model, the stochastic change of the sequence. If we have the generative model, we can do statistical inference based on that. So molecular clock, I will not go into much details about the sequence evolutionary model, but give a quick overview of molecular clock model. Basically, it is a stochastic model of the variation of the rate, mutation rate among the lineages. The most simple model is a strict clock, assuming a constant rate, but then it is unrealistic. So we would like to relax the clock to allow some rate variation, and most commonly used models are LogNormal, gamma, or exponential. We have the generative model, so we can use statistical inference to infer the tree. Bayesian MCMC is the most widely used method, and the most, I can say that the most popular software for molecular dating is BEAST, but it has some drawbacks. It is computationally intensive, and more to that, it is sensitive to the clock model that we choose. So showing here is an example where the estimate by BEAST for one particular evolutionary event can be differed by twofolds by just changing the clock model that we assume. So that saying something about molecular dating, whether we can trust it at all. Because when we look at the real data, what the estimate from molecular dating is very different from what people know from fossil records. And the discrepancy can be as high as twofold. One of the most pronounced example is the flowering plants, where molecular dating, many molecular dating results stay in the very old time comparing to what people know about flowering plant. So showing on the left is the evolutionary tree of the animals, including the dinosaurs. We know, and the right is the plants, and including the flowering plants. What people know is that flowering plants should come to emerge on Earth at the very later comparing to the dinosaur. But the result from beast and some of other molecular dating methods saying the otherwise. The flowering was estimated to be about 250 million years ago, could be as old as the dinosaurs, and is inconsistent with what people know from morphology, from fossil records, from ecology, so on. So, Blue et al. Got the same result when they tried to infer the date of flowering plants using beast, and they have a hypothesis. Actually, maybe beast can be very wrong because the rate distribution also the clock model is not as simple as a LogNormal or an exponential distribution. It can be a multimodal distribution. And when we use a wrong clock model to infer the tree, it can be very misleading. We can get very misleading result, and that may be the case that happened here. So they did an experiment, simulated data, where they fixed the angiosperm date to be 140 million years ago. They assumed a multimodal, like the bimodal clock model here, and they simulate the sequence out of the tree. What they observed is exact same behavior on beast, where it got the estimate to be about more than 200 million years ago, which was pointing out to maybe this is the reason why we see very off molecular dating results. Okay, so we see that the molecular clock, the choice of the molecular clock can be the reason that molecular dating can be misleading. The question is, if the true molecular clock is a multimodal like this, what can we do? So the project goal is to introduce a flexible, a simple yet flexible clock model that free us from the assumption about some particular clock model that we have to parameterize. I will formulate molecular dating as a maximum likelihood estimation problem, and I will develop an efficient algorithm to optimize the likelihood of that model. Okay, so before I go into the details of the method, let's look at the model again. And here, as I mentioned, beast going directly from the molecular sequences to infer the time tree. But we can actually split the process into two steps. The first step, we infer the tree in mutation metric, and then the second step, we just do the dating. The advantage of doing so is that we can rely on very powerful methods for phylogenetic inference that has been developed before, and we can only focus on the second step. In this work, I also assume that the input phylogenetic trees also mean the output of the first step has a correct topology, and but we model the error, the uncertainty in branch-length estimation. Okay, so the model here reduced to having a unknown chronogram as the parameters. We have the mutation rate raw from an uncorrelated clock model that we don't know yet. And then from there, we have error in branch length follow a Gaussian model. So the idea here is we don't want to parameterize the unknown clock model. So we approximate it by a categorical distribution. Or in other words, we approximate the continuous distribution by a discretizing it and show it as a histogram. If we show it as a CDF, it is a step function that approximate the CDF. Okay, so this is the plate diagram showing the when we put the categorical model together with all the other components to have our model. And then the calibration points will define for us the constraint on the valid trees that we can estimate. So we have the constraint maximum likelihood problem, where we find tau, which are the branch length and time units, and the rate categories that maximize the likelihood and respect the constraints defined by calibration points. So when we look at the likelihood function, although it looks simple, we want to speed up the process beyond the optimization beyond using a black box optimizer. So we will use EM algorithm together with block coordinate descent to have an efficient algorithm to optimize the likelihood. So I will not go into too much detail of the EM algorithm here. Basically, in the E step, we compute the posterior. In the M step, we can transform the log likelihood function into a more simple form that we can solve easily using block coordinate descent. And when we look at the function in the M step, it is basically in a weighted least square form that we can solve very efficiently. Okay, so here we revisit the flowering plants. In simulation, as expected, as shown before, bees perform very poorly in this data set. We also compare MDCat, our method, with the other two methods, namely real-time and WLogDate. We see that MDCat has the least error and the estimate is unbiased, unlike WLogDate always overestimate the time and real-time always underestimate it. Okay, so look at the real biological data from Beaulieu et al. Here, MDCat has a much younger estimate for the angiosperm comparing to bees, and this more consistent agree better with the fossil records. Also, we see that MDCat detects the evidence of a bimodal distribution of the rates, as showing here. Okay, so another data we looked at was the HIV. An extension from the dataset simulated by Toh et al. In 2016. Here we look at 12 different clock models and compare MDCat to bees and WLogDate. As expected, bees and WLogDate, when we look at the result from LogNormal, which is the correct clock model that they assume, they have a good performance, but MDCat has similar errors. For the other clock models, we see that the performance of bees and WLogDate degrades severely, but MDCat is still stable. The error is still stable. It is saying that it is robust for different choice of the clock models that we choose. Okay, we also look at the real biological data of the HIV, where we download the sequences from Los Alamos website. And we see that the estimate of most HIV-1 subtypes by MDCat are consistent with previous studies. And we see a very interesting trend when we track the estimated mutation rate through time. We see a drop of the mutation rate in about 2000. That is about the time that the antiviral drugs were developed. Okay, so conclusions and future works. We introduced MDCat, a maximum likelihood dating method using categorical clock model. Thanks to the flexible and simple properties of the categorical clock model, we can design the EM algorithm that can very efficiently optimize the likelihood. For future works, we want to explore the effect of K, the number of categories, rate categories, for the model on the accuracy. And we will incorporate this category clock model into the general maximum likelihood framework so that we don't have to separate the two steps as we did here. Okay, I would like to thank my former PhD advisor, Professor Siavash Mirarab, and my co-author, Eduardo Charvel, and the funding sources. Also thanks the Princeton University for residential postdoc research fellows for the travel funding. And this is my preprint on bioRxiv as well as the MDCat software. Thank everyone for listening.